Weigand, Korea. The last title of last session is bio-geo interaction and CO2 and open session. So there are many sessions inside here. And the keynote speaker for bio-geophysics, bio-geo interaction session is Mr. Maximilian Weigand. So,
thank you. So, hi everybody. So, we were looking, or for the past years, we were looking at using electrical polarization measurements for the characterization and monitoring of crop root systems. So, we've known for a long time now that crop roots exhibit polarization signatures in time-varying electrical fields, but there's been little developments regarding applying imaging techniques to these polarization measurements and using standard geoelectrical or near-surface geophysical equipment to do these characterizations. So, in the first part, I will talk about the signal understanding and some laboratory experiments we did in the last years. And in the second part, I will talk about the ongoing efforts to establish a field EIT monitoring system with the aim of doing this on a field scale now, so applying or doing the root characterization on a field scale in some controlled test fields. First of all, why are we interested in root systems at all? So, probably the most important thing is that roots form an important link between the subsoil and the atmosphere in terms of water evaporation, but also in terms of nutrient and pollutant uptake. So it's always important to improve the understanding of the functioning and the effects of these kind of systems. And in addition, today it's still hard to do root research. So root research is still done primarily by manual labor, excavation. So most of the time roots are destroyed if you look at them. So it would be really nice to have non-invasive measures to look at these systems in situ, in the field, in the laboratory, without having to destroy them. And another thing, even if you're not interested in roots at all, I think it's important to at least estimate the effect they have on our other near-surface measurements that we do. So at least we should be able to specify the effect they have on our measurements on, for example, landslides or these other issues that we have been talking about. So just a quick recap. So we are talking about frequency measurements. So we are measuring the electrical impedance in terms of magnitude and phase values, or real and imaginary parts. We use the standard 4-point spread, and we measure SIP spectra at various 4-point configurations, and then use tomographic algorithms to do the imaging. So pretty similar to EIT, but in the end we have these kind of frequency-dependent signatures that we look at either directly or recovered from imaging results. Our basic understanding of all kinds of polarization effects taking place in the frequency ranges that we are interested in, that is in the frequency bands below a few kilohertz, are basically all related to the electrical double layer, the stable concentration gradients forming at charged surfaces of water, and these electrical double layers can be polarized in external electrical fields, and this polarization we can then measure in terms of phase or imaginary parts. And usually when we talk about electrical double layers, we talk about electrical double layers forming at the grain surface in contact with the surrounding fluid, if we talk about soil or subsurface measurements. But we know that these DDLs also form at all kinds of biomatter, for example, biomembranes at cells in root systems, or if we look at the cross-section of a root system, we can identify multiple structures within these root systems that can potentially be polarized using EDL. So we have, for example, the Casparian strip, which is an iron selective membrane surrounding the central cylinder in the root system. We have the outer root surface in contact with the surrounding soil, and we have cell clusters in the root systems that can also be polarized. And we also have the extracellular space between the single cells that can form electrical double layers. So we think that these polarization measurements give us a direct link to structure and in certain terms also function of these biological systems, in our case the root systems. Based on previous measurements or previous studies and our research, we have formulated 3 hypotheses to analyze and interpret these polarization measurements. The first is, we think that the surface area of electrical double layers that is present in the sensitive area of our measurements is directly correlated to the magnitude or the total polarization that we are measuring. The second one is that if we extract, or that the processes that we are measuring, they take place on certain length scales, and that these length scales can be directly translated or are related to characteristic relaxation times that we can extract from our SLP signatures. And the third one is that these polarization measurements are not only correlated to structure but also to function of the root systems. So we know that the surface characteristics of biomembranes are modified by certain physiological reactions in root systems, by nutrient availability and by stress situation. And we also know that nutrient dynamics are the dominant constituents of ions in the fluid located in the root system. So if we have nutrient dynamics or translocation within the root systems, we think that we can also expect changes in the polarization signatures. So our measurements take place mainly on 3 scales. So we have the small scale, where we try to measure small amounts of roots basically embedded in water, so we can try to come up with some intrinsic signatures. We have the laboratory scale, where we try to image whole root systems in rhizotome containers, and we have the field scale, which is still relatively hard to do if you talk about root characterization and especially polarization measurements. But if we just go to the first scale, the small or single root element scale, if you just measure some roots in small measurement container filled with water, we see hardly any contrast in the conductivity, because prop roots are not often non-woody in nature, so they don't have large resistivity contrasts compared to the surrounding water. On the other side, we see significant polarization responses in the low frequencies that we are usually measuring in the frequency domain. So that's basically supporting our hypotheses that we can use these polarization signatures to gain additional information over just doing conductivity measurements. This is only valid if we measure in water. So as soon as we go to the soil or to more realistic conditions, things change a little bit, but changes in conductivity can then be mainly related to water function. If we go now to the next scale, the imaging of the whole root system, we started with these rhizotome containers filled with water again, and we put quite some root systems in there and imaged them over multiple days. In addition, we used only tap water to create a nutrient stress situation to see if we could also see systematic changes in relation to these quite obvious physiological reactions. If we now look at the imaging at 2 frequencies, basically we see that we see a repetition of the previous results. We see hardly any correlation and conductivity with our extension of the root systems. However, we see nice correlations with the in terms of polarization signatures with the root systems. So we only by their polarization signatures. If we now go a little bit further and look at the time evolution of these polarization anomalies in terms of total polarization, we see a systematic decrease which correlates well to the die-off of the root system due to the neutron deprivation. So we think this is pretty good evidence that we are also seeing these functionality dependence in the polarization signal. We can go one step back, but increase the measurement time to only spectroscopic measurements on one dipole. We can go down to a 7-minute measurement time. We can also see systematic changes in total polarization and in terms of relaxation times over the diurnal cycle. This is for dark and light conditions. We even see slight shifts in these curves compared to the onset of another period, which could also indicate that we are seeing these memory effects that are known to be present in root systems in terms of nutrient storage or photosynthesis. So, yeah, that's basically the state where we are at the moment. So, we know that polarization signatures are related to the physiological reactions. We don't know the exact polarization processes or the processes that cause our polarization, so we have no detailed models yet that can really describe what we are seeing in terms of these phenomenological descriptions. Yeah, we can use common imaging equipment from the geophysical community to do this. However, as soon as we introduce soil into these experiments, we have a superposition of the soil polarization signature and the root polarization signature, and things get complicated. In addition, roots react to different states of water availability, so we also induce additional physiological reactions. And we think one way to tackle these are these numerical modeling approaches. So we can, the idea is if you can model the water content and dynamics in these systems, we can hopefully see anomalies that can be attributed to the root systems. So how do we plan to go one step further to the field scale? So we've been working on an ELT monitoring system for quite some time now, which has been set up at the Seelhausen test field on the Research Center Jülich. And it's a multi-geophysical test site, so we have measurements of root length density, we have GPR measurements, we have above ground biomass measurements. So this is a perfect situation to not only test our ELT equipment, but then in the next step also try to correlate these measurements with our with the biological parameters that are measured there. We have quite some challenges, so ELT measurements require careful setup of cabling layouts of electrodes, and we need electronics at the electrodes for pre-amplification of these small polarization signals. And we have a huge amount of data. So basically, compared to ELT measurements, we have not only magnitude but also phase values for real and imaginary parts, and we measure at multiple frequencies, up to 20, 40 frequencies. So this is a huge amount of multidimensional data that needs to be processed. So we need some kind of automated analysis pipeline. So the system we are working on now consists of 40 electrodes spaced 25 centimeters apart. You can see the electronics. It's quite a challenge to keep the water out of these. We are lucky to be able to do in situ calibrations of the system every day in terms of the electronics, internal strain capacitances, and even some load impedance calibrations, which all allow us to do to develop quality checks for our measurements. So we can give a lot of information to really differentiate between noisy data and data that can be used. One per season we do also inductive coupling calibrations, so we take into account the inductive coupling caused by the cable layout, which has to be done after the setup is completed. Yeah, and we can do one full EIT measurement every hour, which, so the system has been working in an experimental state since April, so we've done more than 1,000 tomographic measurements. We have collected roughly 1 terabyte of raw data, so this is quite an amount to do manually, so we need these automatic polarization or automatic analysis pipelines. And while we don't have a detailed analysis yet, we see, if we look at single dipoles, we see systematic SIP signatures, which are not unlike the ones we measured in the laboratory. So this is quite nice that we are able to measure these smooth SIP curves with this new system in a monitoring setup. And the blue area indicates systematic changes over a 2-month period. So we're confident that these could be related to not only water content or soil-related things, but also to the crops that were growing on the field this year. We see also slight changes in the conductivity caused by water content. That's why it's also important to do really high-frequency measurements to be able to look at the small-scale daily and weekly variations, or even daily variations in water content in these systems. We also did some preliminary inversions. The conductivities primarily show the layering that is present at the site, but we also see quite systematic polarization signatures, which we cannot interpret yet in terms of routing or biological parameters, but they're not completely useless, which is quite nice. So, and in summary, we think that we have a basic system set up that is readily primed for the next growing season. And perhaps one last slide regarding the analysis. So one approach that we took now is to basically split all the analysis of different steps into a single task and group them in task graphs to be able to basically parallelize our whole analysis and distribute it over multiple CPUs and computers in the network. We are storing all our data in databases, so we can do these or do a proper data exchange over the network and manage these large amounts of data. Okay, so to conclude, we are able to monitor or to image crop root extensions and simple laboratory experiments. We've shown that there's some relation to functionality of these crop roots to physiological activity, but we still have quite some challenges in both substrates with the variable water content and the associated changes not only in conductivity but also in polarization responses. We have a more or less successful EIT system established, a monitoring system, and going forward we will do new laboratory experiments in soil and subsoil substrates to get a further signal understanding, and we are also looking into setting up modeling experiments or doing some modeling regarding these root rhizosphere interactions. Thank you very much.
Thank you for your clear presentation. It's time for question. Do you have any questions? Yes.
So, what crops are you monitoring here in the field?
This year's was maize, so large.
And what's the rooting depth of maize?
I don't know. I think 1 or 2 meters.
Okay. And I guess the other question I have is I know I'm familiar with some other people who when they do crop root monitoring, they inject on the plant, so they get basically. I know that you guys are not doing. You're basically injecting the soil, trying to image the plant. Is there a reason for that? I mean, because in terms of the resolving effect there, I mean, like, you know, because don't you want to couple as much current as you have into the plant there, or?
Yes, but you lose your non-invasive manner of the injection. So if you think about this field scale application, as soon as you start injecting current into the plants, you always have the possibility to basically fry the plant from inside. So it's not really non-destructive anymore. Yeah, and another thing is if you force current through the roots, you basically get a totally different polarization signature. So you will basically if you force so this is the central xylem, which basically conducts all the fluid in the root system. So if you force the current flow through this central xylem and then radially out of the root, you will force it through all these layers of biomaterial, and yes, you will get a stronger polarization response, but we are really interested in the imaging of the whole rhizosphere. So at this point, probably more or less counterproductive, because yes, I get more information about one root, but I'm not about the whole rhizosphere.
Okay. Any other question?
Yeah,
Okay.
thanks, Max. I just said, you mentioned the link between the frequency and the length scale, right?
Yes.
But you didn't mention in the end
what length scale you were actually thinking about in terms of your results.
Yes, because I don't know yet.
Oh.
As I said, we don't actually know the detailed processes that we are seeing. So basically, we don't know if we're seeing single cells. It's unlikely because the frequencies are too low for single cells.
That's my feeling.
So if I would have to guess, I would say we are seeing some larger scale structures. So the question is if we are even seeing single roots or just groups of root elements. So it could be just one scale larger, and probably one of the main contributors will be the soil or fluid root interface.
So can I just maybe add a comment on that? So basically, it's all it's driven by diffusion again. I mean, the process is so kind of diffusion coefficient would kind of be the key quantity linking spatial scales of characteristic length and characteristic time. So similar to kind of interpretation of membrane polarization. And having said that, it's actually that it from the order of magnitude, it could be actually the sizes that we have there. We're at the scale of also where we have the roots. Yeah. Although we cover to have a certain distribution. Thanks.
Okay. Any other question? Yes, please.
Oh, such a frequent injection of current into ground, so injecting current to the ground to the root, give any storage to the root system?
So in one of the earlier experiments in rhizotones, we actually checked this and we found no direct variances between rhizotones which basically were measured with EIT and those that were measured without EIT. In principle, yes, you can influence growth of roots and plants by applying low frequency electrical fields, so it's always a matter of current densities that you are basically creating in your measurement system. So we're using voltages of 10 volts maximum, so these are really small voltages. Yeah, so in our experiments, not in general, it's possible, yes.
Thank you.
Okay, we can have more question now because his presentation was a little bit short, so please make more question. Actually, okay.
Just a small one. It's more related to the last question. So in a review, Solomon, which you know, saw that when you looked at biological papers, that plants also have a whole range of electrical signals to communicate within their tissue and so on. So do you have any idea whether you think that what we are measuring is influenced or is in the same order of magnitude of those things or not? What's your opinion about these things?
I think these signals and for example, in biological cells, we have transmembrane potential to range up to a few hundred millivolts, but these are localized. So I don't think we see them if we don't inject or measure directly in the roots. So if we go far enough away, we won't see them.
Okay, no more question? Okay, then thank you very much. And now